So with that, I'd like to invite Dr. Foucher to the podium.
Thank you, Paul. I'd like to thank the organizers for allowing me to speak here. In the next 10 minutes, I will run through the experiments globally, what we have done, why we have done it, and what the context of these experiments was. And also I take some time to get rid of some of the misperceptions that have come through the lay press about our work. This work was done by a large team of investigators in my laboratory. In the first place, Sander Harris, a postdoc in my laboratory. So this work really got started back in 1997 when there was the first incidence of human exposure to fully avian, highly pathogenic H5N1 virus, including the first human deaths related to this virus. And it was very quickly recognized that these viruses were coming out of poultry. There were poultry outbreaks in Hong Kong. The Hong Kong government started to kill all the poultry in Hong Kong, and by that time, after that, it was thought that this outbreak was over, but that appeared not to be the case. The virus remained enzootic in Southeast Asia, and from 2004 onwards started spreading over numerous countries on 3 continents, including parts of Asia, Africa, the Middle East, and Europe. In many of the countries where the virus landed, culling was not the first option. And as a consequence, these outbreaks are still going on today. These viruses have caused hundreds and hundreds of millions of poultry deaths. And on occasions, these viruses have also been transmitted to various mammals, including cats, dogs, pigs, tigers, and also, importantly, they have spread to humans. And from 1997 onwards, the question has been, in the influenza community as well as in the public health community on the whole, can this virus go airborne, and can this virus ever trigger a pandemic? As it usually goes in this type of, when this type of questions arise, the research agenda is set under the umbrella of a variety of organizations, such as NIH, FAO, OIE, WHO, who are putting this question, can H5N1 go airborne, very high on the research agenda. And as a result, various calls for proposals for research were then drafted by the EU, by the NIH, and by many other organizations. And scientists then, of course, start addressing these questions. And here's a list of 5 or 6 publications already that have come out since 2006 where scientists have tried to address this issue for H5N1 virus, but also for other animal influenza viruses that are circulating in the reservoir. Can these viruses go airborne? At Erasmus, we followed these events here on the slide. In 2005, there's been a call for proposals from the NIH to start the Centers of Excellence in Influenza Research and Surveillance Program. There was a broad agency announcement in 2005 that we responded to together with scientists at Mount Sinai School of Medicine. Our proposals were reviewed favorably, and our contract was rewarded in 2006. At that time, we also started to build our special facilities for this type of research. We do not take this lightly. We purposely built ABSL3+ facilities specifically for working with airborne pathogens. We also obtained an explicit permit to work with these viruses from the Dutch government. We applied for such permit from the Ministry of Infrastructure and the Environment, and the ministry was advised by the Dutch Committee on Genetic Modification, and both of these organizations, so the Committee for Genetic Modification and the ministry, judged that under the conditions that we created in the ABSL3+, we could do this type of research with negligible risk to humans and the environment. In 2007, our facilities were cleared by the US CDC as well as our local biosafety office, and this clearance by the US CDC was necessary because overseas laboratories that are funded by NIH need to get approval to work with select agents. And since 2007, we've also been, of course, inspected by the Dutch ministries. The work then started in 2007, and since 2007 we have reported our progress through semi-annual reports to the NIH, but also through the normal routes of publications and conference presentations. But it's important to note that until the summer of 2007, no progress could be reported about transmission because the H5N1 viruses until that time did not transmit, neither in our studies nor in studies by CDC and other laboratories. So what do? The experimental setup is very simple. We infect a ferret in a cage with an H5N1 virus, and we then simply wait whether this ferret will sneeze or cough or breathe, such that the virus would end up in the ferret in the cage next door. And these ferrets are separated by 2 steel grids that prevent direct contact between the animals, but still allow airflow between the animals to occur freely. These animals are not in an open facility. They are housed in a BSL-3 facility, low-pressure HEPA-filtered room. And in this room, all the experiments are carried out in Class 3 biosafety cabinets and Class 3 isolator units when we do animal experiments. And so in many ways, this is similar to the conditions in a BSL-4 lab, except that in a BSL-4 lab, most times the investigators are packed in spacesuits, whereas in our case, we actually wrap our experiments in these spacesuits. So what did we do? We took an H5N1 virus and we took a mutated H5N1 virus, and I cannot give you more details about what those mutants are, and we passaged them in ferrets. And then we see whether those viruses become transmissible by aerosol. And we find that the H5N1 wild type does not transmit, so it replicates in the donor ferrets, but not in the recipients. But if we do this with a mutated H5N1 virus, we can see that in 3 out of 4 cases, the virus is transmitted via aerosol or respiratory droplets. We then take this virus from this ferret, from one of these ferrets here, and try to do it again. And indeed, we can now, in 2 out of 2 ferrets, see transmission again. So this is the first time we have worked with an aerosol-transmitted H5N1 virus. That on itself is an important finding, but on top of that, we now know exactly which mutations and which biological traits are necessary for H5N1 bird flu virus to become aerosol transmissible between ferrets. Now these are the main conclusions of our work, and because our paper still hasn't been published, but the press has picked up on our story, there's a lot of misperceptions what you can and cannot conclude from these studies. Now, the first thing that we've heard many times in the press is that this virus would spread like wildfire if it would come out of our facility. But we do not think that this is the case. So we have shown aerosol transmission, but the efficiency cannot be deduced from our experiment. This is not a quantitative setup. But even if we try to extrapolate a little bit further, if we look here, this is pandemic H1N1 from 2009, and what you can see here is in the lines is the shedding of virus from animals that have been inoculated, and then the bars are titers of virus at different days after infection that have received the virus by aerosol. And we can see for pandemic H1N1 in 4 out of 4 animals, we can see massive virus replication, and this replication starts as early as day 1 in the recipient ferret, and generally all ferrets by day 2 are starting to shed viruses at high amounts. But if we compare this to H5N1, we can see that, first of all, it doesn't transmit in 100% of the cases. Secondly, the virus titers that are being shed in the aerosol contact animals are much lower. And thirdly, a lot of the animals actually do not start shedding virus until day 3, 4, or 5. And so, remember, this is not a quantitative model, but if one would deduce anything from this, the efficiency of spreading, we would have to conclude that this virus does not spread yet like a pandemic or a seasonal influenza virus. The second misconception is that the virus would be highly lethal if this would ever come out. But also here there is some facts to explain. It is absolutely clear that H5N1 is a highly pathogenic virus for chickens. You inoculate a chicken, the chicken will drop dead. But in mammals, this is certainly not the case. In mammals, it is known that if we infect ferrets, for instance, with very, very high doses, 10 to the 6th of virus, and put it straight down into the lungs of the animals, yes, the animals will drop dead, and they will do so by around day 3. But if we inoculate ferrets or other mammals via an intranasal route, the animals generally do not get sick at all. They might get a little bit of flu, but they certainly do not drop dead. Now, the virus that we have used does cause disease when we put it via aerosol, when we put the virus in the nose at very, very high titers again, right? 10 to the 6 in the nose, we get some neurological symptoms, and it's known that this is very typical for the virus we use, but not a general observation for H5N1. But it's important to note that the mutant virus only causes disease in 1 of 8 animals if we inoculate at this very high dose. But if we now look after aerosol transmission, we actually see no disease, no severe disease at all in any of the 7 animals that received virus via aerosol. So this virus, although it is highly lethal to chickens and highly lethal if you put it directly down at high titers in the lower respiratory tract, it is certainly not highly lethal if ferrets start coughing and sneezing to one another. It's also important to note, and I'm sorry that my slides have shifted going from Mac to Windows here, that when ferrets are pre-exposed to seasonal flu, they are fully protected from developing severe disease. So if we compare that to humans, and you all have been infected previously with seasonal flu, it would be unlikely that you would have no cross-protection against a virus like H5N1. And so very few individuals would actually develop severe disease, but most of them would actually be protected by cross-protective immunity. So I'm going to conclude that if you ask me, should this work be done? The answer is yes, but we have to assure appropriate biosafety and biosecurity conditions. And I think that we have met those biosafety and biosecurity conditions very well. And why we would be doing this was, first of all, of course, in the interest of fundamental science. This is the first time ever that we have made an animal flu virus transmissible via aerosol, and this will really allow us to better understand how viruses are becoming aerosol transmissible, and therefore will aid in our understanding for pandemics of the future. Secondly, we think that there's a direct implication for public health. First of all, we try to prevent potential pandemics by putting out warning systems to the countries where H5N1 is circulating. And until today, or until a couple of months ago, the expert advice to those countries about the public health risks of H5N1 were not unanimous. And now they are going to be more unanimous now that we've shown that H5N1 can become airborne. Secondly, because we now know the biological traits of these viruses and the mutations that are associated with those biological traits, we might be able to do better surveillance. We might be able to study viruses in outbreaks and to see whether some of these mutations are already occurring. And indeed, we see over recent years that the mutations that we find and that Yoshikawa found are becoming more prevalent in the field, and that is not good news. If we fail to prevent a pandemic, then we might still be able to handle it better when it hits. And we can do so by testing vaccines, antivirals, clinical management, better with viruses that have the proper biological traits, viruses that are transmitted via aerosol. Now, you've all noticed that there has been an enormous discussion in the public health field, in the lay press, and because there was so much controversy, we and Yoshikawa's group and all other groups that did this type of work or had the capability of doing this type of work called for a moratorium until the end of March. And the reason to do this was to give more room for the discussions as we are doing here today, hopefully. There are clear challenges and there are clear opportunities from our research, and those need to be handled as well as possible without getting into the wrong conclusions by acting too quickly. And it is important, I think, that everybody who is making decisions about this work know the details of our study. Right now, there are too many misperceptions about this work simply because the work cannot be evaluated by everybody involved in making the judgments, and therefore I think it's important to publish this work in detail. I'll stop there.
Thanks, Rod. Next question.
Roxanne Kamsey with the news section of Nature Medicine, and this question is for everyone. I was curious to know whether there are any security measures being taken to protect the data right now? Because we live in an information age, I wonder if there are people who aren't waiting for a publication but might be trying to access the data as it's emailed or discussed on the phone or anything like that.
Ron, do you want to? I mean, one of the problems with security is you don't discuss it in an open forum, but maybe you can be reassuring at least.
That's right. And so one of the problems is, of course, that scientists are usually forced to be open about everything they do because we're working with taxpayers' money. And so did we do that this time. But certainly there have been cleaning operations, so to speak, to get our computer systems cleaned up. ICT security, as you all know, is just not doable. So any good specialist could hack different systems. The manuscript has been shared with NSABB members, journals, and amongst the authors. We're doing everything we can to protect the manuscript. We already did everything we can to protect the virus from getting out of our facilities. And I will leave it with that for now.
Thank you.
You know, I'm speaking personally now, not on behalf of the NSABB. But I am one of those influenza researchers that has been very supportive of the work that Ron is doing and thinks that that type of research, as Tony just laid out, is very critical research. Our point has been how it can be done safely. This has been a misinterpreted comment, but I really believe that of all the agents that could accidentally leak out of a laboratory, for example, there is none like influenza. Influenza is the equivalent of having a screen door on your submarine. Once it's out, it's gone. Smallpox, any SARS, any number of viruses, it would be a horrible tragedy if they ever were to leak out, but we could contain them and it would not see a global pandemic. So I think that, again, issues around safety, issues around who has access to the virus. I've not personally been worried about somebody in a cave somewhere, which has been characterized often as the terrorist approach, is the one that's going to do this. I worry about the garage scientist, about the do-your-own scientist, about the person who just wants to try to see if they could do it. And today you may say that's difficult, but in fact it's quite remarkable to see what's happening out there with sequencing, et cetera, and so forth. And so I just point out that I think all of us really have at the heart, we need to know much more about this. The real risk we have today is Mother Nature-made pandemics. But there are legitimate concerns to make sure that we don't have a problem. 1977, H1N1 was an accidental release, no doubt about it. We don't want to see that happen again with a virus like this. What we're trying to do is come to the middle. How to do this research safely, how to do it in a way that furthers all the points we've just talked about, and at the same time, make sure that we don't create our own problems here. And that's why I think we're asking for this pause to think about this and to move forward in a positive, proactive way.
So one last question, Petra.
Well, I suppose it's... Can you hear me? I'm too short for the microphone. I was just thinking, I've been listening to this, and actually I feel that this is quite a positive situation. The peer review system worked. It caught the papers. There was a system came into play that allowed people to discuss it. So personally, I feel this is a quite positive thing. Okay. I think there's a bit of concern about how we then identify who are legitimate researchers who can access the data in order to further the needs of mankind. But I think what this and so many other cases show is actually journalists in the common press have an awful lot of responsibility here. It's easy for them to knock the scientists doing this work, and it's easy for them to use terms like doomsday bug. It's lazy, sloppy reporting. And I think you should be bringing just as much effort to bear on making the journalists behave responsibly as you do to making the scientists behave responsibly. So.
You should make the journalists be as responsible as you.
So I'm going to